By making use of enzymes found in the digestive tract, MIT engineers have devised a way to apply a temporary synthetic coating to the lining of the small intestine. This coating could be adapted to deliver drugs, aid in digestion, or prevent nutrients such as glucose from being absorbed. In a study conducted in pigs, the researchers demonstrated that they could use this approach to simplify the delivery of medications that normally have to be taken multiple times per day. They also modified the coatings to deliver the enzyme lactase, which helps people digest the milk sugar lactose, and to block glucose absorption, which could offer a new strategy to treat diabetes or obesity. The lining consists of a polymer made from dopamine molecules, which can be consumed as a liquid. Once the solution reaches the small intestine, the molecules are assembled into a polymer in a reaction catalyzed by an enzyme found in the small intestine. The MIT team began working on this project with the goal of trying to develop liquid drug formulations that could offer an easier to swallow alternative to capsules, especially for children. Their idea was to create a polymer coating for the intestinal lining, which would form after being swallowed as a solution of monomers. They took their inspiration from nature and began to experiment with a polymer called polydopamine (PDA), which is a component of the sticky substance that muscles secrete to help them cling to rocks. PDA is made from monomers of dopamine, the same chemical that acts as a neurotransmitter in the brain. The researchers discovered that an enzyme called a catalyzed could help assemble molecules of dopamine into the PDA polymer. Catalyzed is found throughout the digestive tract, with especially high levels in the upper region of the small intestine. In a study conducted in pigs, the researchers showed that if they deliver dopamine in a liquid solution along with a tiny amount of hydrogen peroxide at levels recognized to be safe, catalase in the small intestine breaks the hydrogen peroxide down into water and oxygen. That oxygen helps the dopamine molecules to join together into the PDA polymer. Within a few minutes, a thin film of PDA forms, coating the lining of the small intestine. Once the researchers developed the coating, they began experimenting with ways to modify it for a variety of applications. In this study, the researchers showed that the coating lasts for about 24 hours, after which it is shed along with the cells that make up the intestinal lining, which is continually replaced. For their studies in pigs, the researchers developed the solution by endoscopy, but they envisioned developing a drinkable formulation for human use. The researchers are also developing other alternative formulations, including capsules and pills. The researchers performed some preliminary safety studies in rats and found that the dopamine solution had no harmful effects. Their studies also suggested that most or all of the dopamine molecules become part of the synthetic coating and do not make it into the tissue or the bloodstream, but the team plans to do additional safety studies to explore any possible effects the dopamine may have.